And I think with that, I'll let Michael begin.
Oops. Thank you, Ellen. Thank you all for joining while I try and find my laser pointer, which I obviously unprofessionally should have done a moment ago. So thank you all for joining, and thanks to, I believe that Epicipher is going to be running a number of webinars this year. They asked me to do the first one because I think they wanted me to give the sort of the philosophy of the way that we are doing the stuff that we do, also to give a flavor of how we have applied the technology that we use. And so I chose a title that reflects, that sort of feeds off the philosophy that we've had within the company. And this is, we're a technology company, but tech is not much use unless you actually derive some interesting biology out of it. So it's seeing the application of the tech is a lot of fun. We get to play with it a lot before you do, of course, because we've got the R&D group, which then feeds into our collaborations. And so with that, we get an opportunity to sort of see, well, is this tech fit for purpose? So I love this quote. I actually heard it first last year, embarrassingly, considering it's such a great encapsulation of our philosophy of things. You know, essentially that progress in science depends on tech, which then informs, because the tech informs the questions that you can ask and the data that you derive. So what have we done with this? If you remember nothing else from this talk, I think I always like to put a take-home message at the beginning. And this is a lot of work that's done in epigenetics, particularly over the last few years, although the field is really changing, and particularly with regards to the cryo-EM people, and I'll come to that at the end of the talk. But it is, we've discovered and learned, and it was known from the very beginning, to be honest, it was that nucleosome context versus the more commonly used peptide-based approaches really do refine the experimental data. They change what you see, and what you see is probably more representative of what's really going on. And of course, this informs the underlying biology, because it's a great way of thinking about it, that in every experiment, you want to be using the most physiological opportunity or technology you can. So this is a slide that I've been giving a version of this for about the last 15 years since I was a PI and going on the PI market. And essentially it was to inform these new potential colleagues of mine, what is this epigenetics thing or chromatin thing that they're bringing into their department. And so essentially we have the DNA wrapped up with histones into nucleosomes, higher order chromatin structures, and then this is encapsulated or packaged into the genome. The repeating recurrent structure is the nucleosome, and this is the Luger-Richmond structure, which is just absolutely spectacular when we saw it first. It gives, because it informs so much of potentially what's going on, and the numerous points protein DNA contact is just stunning to imagine that this sort of shooting. I'm not sure. Oops. I apologize. There's an issue here. So Ellen, can you still hear me?
Yes, it did like go in and out for a second there.
Okay, sorry about that. Yeah, something weird happened in the phone or something. So anyway, we've got this recurrent structure, which is the nucleosome, and I just call attention to the way that we visualize the nucleosome. So the way that it was seen, you have got these beautiful, about 70% of the mass of the histones is wrapped around by the 147 base pairs of DNA. The tails, the extensions of the histones, were not visible in the original structure, but to give a relative idea of scale, they were showing extending from the structure. A lot of us started to think about them in that way, and I'll inform that it's actually not that way, and that changes everything, and we'll see this over the next few slides and over the course of the talk. Over the next few years, we were extremely focused as a field on the identification of post-translational modifications. And so to show how good we were becoming at it, the tails were painted with modifications. And so we would extend the tails and then show that, like, how busy they were becoming with all of these modifications appearing. And I almost certainly said a statement similar to this down the years, and I was entirely wrong. And essentially it says that note the tails protruding from the structure, because they are in the model. And in this way, they're easily available for recognition and modification. And the problem is that statement is factually totally incorrect. The tails are actually engaged with the nucleosome. And so therefore, anything that wants to get to the tails there's a negotiation that takes place to get that. And we'll see why in a few moments in a slide. But Remember the players, and epigenetics and chromatin biology is extremely complicated. You never see proteins wandering around on their lonesome. So you don't have a given enzyme that is just an enzymatic catalytic site. It's also, they are large proteins and large complexes. And so they can be summarized into groups or classes. So we have writers which add modifications. We have readers which read them, the post-translational modifications. We have erasers that remove them. And then we have remodelers that move complexes or remove the nucleosomes around, take them out of place to create open structures for DNA access, or indeed swap out certain histones that are there for other histones that look similar, known as variants. These things do not act on their own. There's generally multi-subunit complexes which can contain multiple reader activities, plus indeed multiple other nucleosome binding activities, including DNA binding, acidic patch binding. Plus, you can have what appear to be completely opposing activities within the same complex. So it's extremely complicated to dissect this biology. It is, however, also much more than simply an academic pursuit. There's multiple drugs which are already inside patients, and there's multiple molecules going after other epigenetic components that are in preclinical development and seen in hundreds of ongoing clinical trials. So, coming back to the nucleosome again, I've mentioned this concept of the tails being inaccessible. So if you look at if you look at the sort of classical density structure, you can't really see why this would be the case. And again, you actually see the tails extended. You see here the particularly long histone H3 tail. So remember, there are 2 copies of each histone within the nucleosome, and this becomes quite important later. Looking at it this way, you can't really understand why the tails would be engaged with the nucleosome surface. But it's completely obvious when you actually look at the charge. So here you have the negatively charged DNA and the positively charged histones. Therefore, the two of them attract each other. So therefore, this tail is not free floating. It's actually engaged with the DNA and the acidic patch. This is itself. Sorry, go on.
Sorry, I didn't mean to interrupt. It's echoing. Is there a way for you to make the slides full screen? Because some people are having an issue. They say the slides are small. This slide is full screen, actually. It's taking up the entire window.
Okay.
I'm not quite sure. Maybe we'll just unshare and share again. See, technical problems, people, when you're doing webinars. Let me just unshare this.
For some people, you may also be able to click the display button in the panel at the bottom of your screen, and you may be able to change how the windows are viewed. Because for me, Michael was also, his slides were taking up full screen. Now I'm full screen.
Okay, now I'm going to share again. Sorry, just in case there was a problem. Entire screen. How about now?
Yeah, I mean, it always looked fine for me, but other people were having an issue. So I would say if it's still an issue, I would say go to the display. Okay, it looks like it's working now. Thank you.
Okay, all right, let's go back to this. So as I was saying, you can see, you can't really understand just looking at the identity of the factors, but when you look at charge, you can understand what's going on. And so we have the positively charged histone tails associated with the negatively charged histone, wrapping DNA, plus also the acidic patch and nucleosome surface that turns out to be really important for protein-protein interactions. Sorry, let me just bring my laser pointer back here again. So, but that is itself potentially regulatory. So for example, this positive charge on the tails is mediated by a selection of positively charged amino acids, lysines and arginines in particular. And you can actually neutralize the charge. And in fact, there's entire modifications which part of their role is to neutralize charge. They're also seen for binding, but they also act to neutralize charge. And the most common ones that would be understood by people are acetylations of the lysines and citrullination of the arginine. So this can, of course, change tail accessibility. So there's a large dynamic thing. Default, unmodified, associated, but acetylation opens up the tail. And opening up the tail turns out to be really important. So everything that engages with the nucleosome, I told you a moment ago that there's always complexes. So everything that engages with the nucleosome is multivalent. There is never one single point of contact within a complex. So over the last few years and in recent papers with some phenomenal collaborators where we've played with their technology, over the last few years, we've been exploring this multivalency. And so we have, for example, a double reader, a PhD and a bromo, binding to 2 modifications on the H3 tail. and that's K4, a methylation and an acetylation. So you'll see the interplay between modifications through this talk. We even have a triple reader that was done with the Shalaterford lab, and this is 2 binding events on the H3 tail and 1 binding event on the H4 tail. We've also seen things where we're binding DNA, a post-translational modification, and DNA at the same time. And so this is work that we've done with the PWWP domains, with SGC, or with Chao Lu, Dave Alice, and Jacek Majewski's group. We've seen the impact of charge neutralization directly. And this is work that we've done with the Strahl lab and the Musselman lab. And that's with regards to both binding activity and modification. We've also seen that the things for binding both post-translational modifications plus the acidic patch plus DNA, so 2 non-specific, which you would class as non-specific nucleosome surfaces. And this is the binding of K119 ubiquitination by DNMT3A1 UDR, ubiquitin determining region, or the recognition by a PRDUB, which is a de-ubiquitinating enzyme for K119, essentially how it is controlled towards its target. And then finally, we've got proteins that appear to bind everything. And so this is a really interesting protein from tardigrade, work with Jess Tyler's lab, damage suppressor protein, which we've shown over the last year to have binding activity towards the acidic patch, DNA, and the histone tails. And this is important for its biology in that it manages to coat the entire genome. So we've been busy. So how have we managed to do all this? We've done it because we've had access to nucleosomes, and lots of them. So the simplest thing when we started Epicipher a lot of years ago now, it feels like forever, but was with the ability to make unmodified nucleosomes. So this is 147 base pairs of DNA wrapping 8 proteins, 8 human proteins. When we learned how to do this, we could learn how to do things like variants or oncomutations, particular mutations that are associated with cancer. The centerpiece of the portfolio, and the thing that actually makes everything possible, is the ability to these things called designer nucleosomes. Designer nucleosomes contain post-translational modifications. They are fully defined in our hands. We don't do enzymatics, we don't do analogs, we do the modifications themselves. And we've now gone after multiple classes. I think most recently, we're now working on ADPR as an example, but ubiquitination was something we were working on some years ago. But we now have the ability to put pretty much any modification we want at any place, plus combinatorials, and end up with a fully defined nucleosome, which has a modification at a given place. And you can create combinatorials, you can sort of do all sorts of interesting things. Most recently, heterotypics, which I'll come to in a few slides. The other thing we can do is barcoding. And this basically means that we change the wrapping DNA around nucleosome A or nucleosome B, such that we can then pool them together and then distinguish what they're doing in reactions. We've been capable of doing this within Epicipher because we've been extremely generously supported by the NIH, to which we are immeasurably grateful. And this is through the SBIR program of the NIH. And basically, we've gone to multiple agencies, and they've given us the ability to make multiple nucleosomes of various flavors, to develop technologies to use them, and to basically apply them to certain disease states. So one of the things we did, the first thing, in fact, we did when we started Epicipher, when we had access to our first pool of nucleosomes, was to actually say, well, we wanted to support and fix CHIP. And so this is a standard CHIP experiment where you take a cell, you have a mess of chromatin, you basically cross-link it or don't, you blow it open, you create a mononucleosome suspension, and then if you're going after a post-translational modification, you add in an antibody, and it goes after its target, you wash like crazy, and then you sequence or amplify the associated DNA that you've enriched. So that's a chromatin immunoprecipitation, a CHIP experiment. We all knew at the time that CHIP experiments were flawed in that even if you managed to enrich for something, you were never absolutely certain that you were enriching specifically or efficiently. So to figure out if this was happening with various antibodies, we created spike-in panels of nucleosomes because we were able to make them. So the first thing that we made, the first large thing we made within the company was the K-Metstat panel. So this is lysine methyl status, and it is unmodified or mono, di, or trimethyl at H3K4, K9, K27, K36, and H4K20. Anybody who's been working in the field for more than 5 minutes understands that there's loads of modifications that are in here that are associated with various parts of biology. Plus, there's others that aren't discussed that often. And the reason was we didn't just want to support colleagues who were existing when we started the panel. We wanted to enable colleagues in the future saying, look, I know you're currently studying K4 trimethyl, but you might want to consider K4 di and mono as well. And so over the last few years, what we've learned is that each of these has their own unique biological or genomic distribution, which is almost certainly informing biology. And I'll show you an example of this later. 16 nucleosomes, spike them into a chip experiment, and then ask, what was the recovery of these? I have an antibody I've just paid for. I believe that antibody is going after K4 tri, so it should go after this nucleosome and it should ignore everything else. And so therefore we said for an antibody to pass, it has to show less than 20% cross-reactivity and greater than 5% recovery. Both metrics are important to us. Specificity is obviously why, but efficiency is also important, because particularly if you're going after low input applications or very precious tissues, you want your antibody to be pulling for as much as conceivably possible. And in fact, we've seen efficiencies ranging all the way from 90% down to 0.1% or 0.01%. I know which antibody I'd much rather be working with. So the question was, when we started getting the data, and some of this was published some years ago in Molecular Cell, another major study is currently in preparation with a lot of the data on chromatinantibodies.com. Go take a look at it. It's pretty terrifying. So essentially, we had a large panel of lysine methyls, 16. We had a comparable panel for lysine acyl, about 24 nucleosomes, I believe. So here we're just looking at a set of around 400 commercial antibodies, which were brought into the lab to actually check, well, okay, what is your specificity and what is your efficiency? And within that data set, the failure rate was 70%. Pretty terrifying. And so we started showing it to colleagues. And of course, as we all believe, we say, well, we're different. We went after the antibodies that actually are better than this data set. And so, of course, we went after the antibodies that our colleagues are working with, because that defines better antibodies, doesn't it? And so we took a look at those. And so we took the top 10 most cited targets and the top 3 most cited antibodies per target. That's a data set of 30. 24 of these antibodies were in the data set, and the failure rate was worse. It was 80%. It turns out these antibodies are very, very good at one thing. They've been largely selected for their recovery, in that they consistently yield data. And if they consistently yield data, I can consistently do experiments. But sadly, it's not necessarily the right data. Okay, we are not talking about a failure rate of 21% cross-reactivity or 4.9%. So it's actually sometimes profoundly problematic. So this is chip data. We now mostly do cut and run, but this was all chip data. Here we're looking at 3 different PTMs, K4 mono, K4 di, K4 tri. So K4 mono is in green, di in blue, tri in red. And so within each box, we're looking at 2 different antibodies. But here what we've done is we've set this antibody to K4 mono to 100% and then asked, well, what is it cross-reacting with? What you see is it's doing a pretty good job, as is the one beside it. It's got a bit of cross-reactivity to the unmodified. But what I want to call attention to is the purple bar. Purple bar is the efficiency score, which is over here. And this is a log scale. And so this antibody is 90% efficient. This antibody is 0.1. Therefore, you can see why this antibody fails. Monos, dis, tris. The biggest problem for us lay inside the tris. And you can see it's profoundly problematic here, because these 2 antibodies are actually, they're to K4 tri, are cross-reacting 60 to 80% with di. It's worse than it looks, because if you actually look at the PTM abundance inside the cell by mass spec data sets, di is more abundant than tri. So you're getting profound cross-reactivity with a target that's much more abundant. Also, this shows up in the chip data sets. Here's 2 good antibodies to mono, di, and tri. These are the cross-reactive ones. So what you immediately see is that their K4 tri signal is strikingly reminiscent of the K4 di signal. What they're actually doing is seeing the K4 di. This is what a clean antibody is supposed to look like. And at the end of this study, the Molecular Cell paper some years ago with Alex Ruthenberg and Scott Rothbard, what terrified us the most was that these antibodies were basically what the field was using, over like a couple of hundred to 1,000 citations, whereas the actual real signal had never been published. So this, of course, informs the problems you're going to have with the biology. What was the reason why? It was tracked back to the nucleosomes. And so this is a chip experiment with spike in nucleosomes, but this is the gold standard approach to screening an antibody, and it's using a peptide array. So you can make these really, really complicated peptide arrays, which contain lots of PTMs, and there are sort of things that look like them. The only problem is if you yield the specificity data from this platform and this one, there's no relationship between the 2 data sets. So in other words, you were creating lots of specificity information from the peptide array. The problem was that it wasn't reflective of what was going on inside the chip experiment. So you were basically wasting your time. If you want to know if an antibody works in chip, you need to check its specificity in chip. And this is something that's driven us all the way along. When we're actually qualifying an antibody for an approach, we want spike-in standards, which represent the target, and we want them in the approach that we're actually using. So over the years, we've sort of fallen out of love with chip for a variety of reasons. And one of the things for which EpiCypher is now best known is for cut and run. And so we released cut and run and cut and tag, and we have additional sister techs in-house, all under the Katana umbrella. It takes a different approach. It's derived from technologies that were developed by Steve Helikoff and Uli Lemley. It's an immunotethering approach. So I'm just going to particularly talk today about cut and run. Here you take your cell that you were blowing apart a moment ago for chip, but now you keep it intact. You have this cell or a nucleus, which is permeable, and they're permeabilized and immobilized onto a solid support. So they're generally Conné beads, although others are available. You then basically take your antibody to the target of interest. You add that to the cell mix. It basically goes inside because it's permeable. It finds its target in situ and it binds to it. So in this case, it's a PTM. You then add in protein A GMNase. The protein AG finds the antibody, which now the antibody has now localized MNase to that location. And now you activate the MNase. And what happens next is the thing that makes all of this worthwhile, is because this complex, this diffuses out of the nucleus. So now your signal is out in the supernatant and your noise is inside the cell. What you then do is you hoik the noise out of solution by basically putting it onto the magnet, and then you create libraries out of the signal. This shifts everything in your favor. We thought it was too good to be true at the beginning as well, but it turns out to be transformative. It changes the signal noise ratio, which is seen in chip, dramatically different in cut and run or cut and tag. And that basically allows you to do a great number of things. Since signal noise has shifted in your favor, you now need fewer cells, reduced sequencing depth, etc. We've done numerous protocol improvements, and we've made all of them available and to anybody through our collaborations, through our in-house development. We've made protocols for sample prep and transfer, etc. We've shown the suitability. My first love is PTMs, but we've shown that it's also suitable for transcription factors, readers, ATPases, and I'll show you some additional modifications we've made to it down the years as well today. And the one thing that we're so happy we've done in-house, because we generate so much data, is that we've actually automated as well. This technology called auto cut and run, which we're running 96 well plates and basically doing them all day, every day, and yielding data in a cost-effective manner. Because we had access to cut and run and cut and tag from such an early stage, we felt that we had the opportunity, and in fact the obligation, to standardize it from the very beginning. So we came back to our K-Metstat again. The footprint is slightly different to make it compatible with the cut and run and cut and tag approaches, but you have a 5' and 3' linker, and then these are immobilized onto biotin beads. So now you have your nucleosomes on beads and you have your cells on beads. You just mix them together at a defined ratio such that you're sacrificing about 1 to 5% of the sequencing depth to the standards. But this allows you to monitor the experiments. It allows you to monitor sample quality, etc., to define new antibodies and to monitor how they're doing in subsequent experiments. When you have access to this, you can finally go searching for antibodies that work. For the first part of this talk, I emphasized the bad, but now let's emphasize the good. There are antibodies that exist that are capable of going after PTMs and doing so with specificity and efficiency. And those are the reagents that we should all be using to basically explore the biology that sort of comes out of those. So here we're looking at an antibody to K4 mono, di, and tri set to 100%. And what we're looking for is a cross-reactivity of less than 20%. And so you see some of these antibodies are absolutely magnificent. This is a data set from about 1.5 years ago. I think we might have now found within Metstat an antibody to each of the targets. So therefore, these are the antibodies that we want to be using. We can also control not just for specificity, but for efficiency. And that's basically the one way to check that is just use fewer and fewer cells and see if you yield the same peak structures. This is a new antibody that we've created to K36 di. That's at a standard cut and run input of 500,000 cells. But you can see we can go the whole way down to 5,000. We are essentially recapitulating the same peaks. We're not losing anything. This is a magnificent reagent. And again, if you're doing low input applications, reagents like these are the things that you actually want to be working with. Once you've got access to all these antibodies, you can start to ask, okay, the previous antibodies I was using couldn't tell the difference between methyl states or cross-reactive across residues. But now that I've got antibodies that work, what does the biology look like? And it's absolutely mind-blowing. I mean, we've seen so many differences between the addition of a single methyl group, so many differences in terms of their genomic distribution. Here is the example we sort of love to show is K27 methylation. Mostly talked about through the PRC complex, K27 trimethyl, associated with repression. But the PRC complex is capable of mediating mono, di, and trimethyl. And when you look, the trimethyl is not the only state that's sitting in a given location. You can see immediately the three are not piled up on top of each other. There is a specific distribution for tri, di, and mono. Not only is there a specific different distribution, they're associated with different genomic features or indeed the activity of the genome. So K27 trimethyl is generally associated with repression. So you can actually look at this by asking, well, is it associated with active genes or repressed genes? So the one way of looking at, here we're looking at, we've got 40,000 genes, or 20,000 genes, I believe, lined up, transcription start site to transcription termination site, and then binned by transcriptional frequency. So red is the most highly active, dark blue is not transcribed or repressed. And what you see is K4 tri is associated with promoters, transcription start sites, and it's associated with active genes. K27 tri, you flip it. You now see it across the entire gene body, but it's associated with the most repressed locations. But as you see, as you go from the tri, the di to the mono state, there becomes a flip. Now K27 mono, product of PRC, repressive complex, is actually associated with activation. All of this we have seen with the other PTMs that we're looking at as well. So we've got access to a lot of PTM maps that we're now integrating, trying to understand what they all do. They all show their own specific distributions. Some overlap with other things. Some seem to be pretty unique based on what we've looked at so far. And I remind people, these PTMs do not exist to recruit antibodies. These PTMs exist in vivo to recruit downstream factors like reader domains. With cut and run, we do have access to pretty much the entire genome, even though it is a three-dimensional native chromatin structure. Here what we're showing is that we can actually yield data from euchromatin, being K4 tri, heterochromatin, K27 tri, and this is facultative heterochromatin, and then this is repressive heterochromatin, the hardcore stuff, constitutive K9, K9 di, K9 tri. So we've basically shown this. We've also mapped onto the new genome builds that are available, for example, telomere to telomere. We're able to look at centromere structure, et cetera, which is absolutely amazing. So let's flip from this. Now we've shown that we've done all this work with antibodies. We now have the ability to map where everything is in the genome. But what we're also interested in, well, the PTMs exist for a reason. They presumably exist to basically as parts of biological pathways. And so therefore readers are presumably involved. So PTMs, we all know for years, recruit reader domains. And we have become extremely good at identifying. So methylations, for example, are big factors. Here's K4 methylation, particularly the tri-state recruiting various family members to it. And these obviously are able to now recruit the complexes they are sitting in are recruited to regions that are marked with this PTM. However, I mentioned earlier, acetylation also neutralizes charge. So acetylation is known to recruit bromodomains, but I think a lot of us probably forgot what was first identified when acetylation, even before readers were even known about. Acetylation is known to neutralize charge. Coming back to this structure again, we have the nucleosome and now the charge patch, or the charged sort of distribution across this, positively charged histone tails, negatively charged DNA. So these two interact. But you understand how they're interacting. It's through basically the various modifications, but you can neutralize that modification by basically acetylating it. This turns out to be really important for biology. So there's the simplest way of work that we did some years ago with Kanish Jan and Brian Strahl and others, and it was published last year in eLife. Essentially what we have here are three reader domains. They're PhD fingers, and they bind K4 trimethylation. That was worked out. So that is their binding to K4 trimethylation in a nucleosome context. Except if you add acetylations to the H3 tail, K9, K14, K18, what you now immediately see is that these PhD fingers absolutely adore that. They're not binding to the acetylation directly, but they're actually feeding off it. And what you can immediately see is that this nucleosome is a much better template because presumably the tail has been neutralized, removed from the surface, and now the PhDs have much easier access to it. You can see this on nucleosomes, and there's obviously interesting biology here, but you cannot see this on peptides because peptides are constitutively available. And so here we are looking at the binding to K4 trimethyl peptides. The addition of the acetylations along the peptide tail make absolutely no difference. The other thing we normally see, by the way, is this additional data. We see it binding to lower methyl states. Normally what happens when we move to nucleosomes is we see gains like this and losses like this, where it doesn't appear to be able to go after the lower methyl states. This has been known about for readers for quite some time, but work that, what Kanishk said was, well, okay, it's actually driving the reader binding, but how about enzyme activity? So we have this complex MLL1, it's a pentameric complex, which was made for us by Michael Cosgrove. Made this complex and then basically threw in various nucleosomes and asked, well, how does the methyltransferase complex go? It goes straight for K4, and it can catalyze the monodi and trimethyl states. But I just want to call attention to a few little things. Just this block over here. This is the activity of MLL. It's an endpoint methylation assay towards an unmodified nucleosome. But this is its activity towards a nucleosome where the H3 tail contains acetylation at K9, K14, and K18. The tail is more accessible. The tail is a better enzymatic substrate. You can see it in an endpoint assay, and you can also see it in the time course or dose dependence assay. The acetylated target is a much better substrate for the enzyme. Question, why? Maybe it's because this is an enzyme complex. Maybe it's because it's just binding better to the nucleosome. The answer is, well, that's not the case. In fact, this is what's known as an alpha decipher assay we use internally. We'll come back to this in a few moments. But the idea is, if I'm a stronger binder, I'm shifted to the left. But the thing is, none of these curves are really shifted in any direction at all, whether I be an unmodified nucleosome or a nucleosome that contains acetylation. So MLL doesn't really, it binds to nucleosomes, yes, but it doesn't actually prefer an acetylated nucleosome. That's not the reason why. What we particularly wanted to be certain about, there are two H3 tails inside each nucleosome. And so what we wanted to be certain was, well, if I acetylate one tail, I should remove that one from the surface, but the other tail should remain in place. Prove to us that it is the acetylation on the given tail that is creating a better substrate for that. So to do that, we have to learn how to make heterotypic nukes. We normally don't talk about how we do some of these things, but in this case, we were so proud of ourselves that we decided we were going to show people. And so here, to make this happen, it's based on a technology that was first done by Beat Feertz. Essentially, what we do is we have two peptides, which are defined with different methyl or modification status, and they're linked by a bridge. We ligate these peptides onto a histone core. Essentially, what we have now are two histones that are joined together, in this case, H3. We call this a bridged heterotypic dimer. We then assemble this into an octamer, assemble that into a nucleosome, and now we have a bridged heterotypic nucleosome, and then we cut off the bridge. And what we're left with is scarless, where the two tails are differentially modified, and we can move things around within that context. Just want to call attention to this over here. Here is where we now have differentially modified the two tails and given it to MLL and asked MLL to play with it. Here we have one tail that is unmodified, and the other tail contains tetraacetyl. So there's 4 acetylations inside the nucleosome, but they're all in one tail. The other tail is unmodified, and it's associated with DNA, and therefore that is the substrate. That's its substrate capability with MLL complex. However, this is the alternate approach. Now we have one tail contains K4 tri to block it from MLL, and the other tail has 3 acetylations, K9, K14, and K18. K4 is available. The tail is neutralized, the tail is removed from the surface, and this is exactly what MLL is looking for. It's much more accessible. So it is direct charge neutralization on a tail. It's not simply having acetylations inside the nucleosome, it's actually having them at the right location. You can see this inside actively cycling cells as well. Methyl and acetyl travel together. Higher acetylated forms, or the higher the acetylations you have on the tail, the more likely you are to see methylation in those locations. And you can see this is work that was done with Nick Young in actively cycling cells. You can also treat these cells with sodium butyrate. This is middle down mass spec, sorry, I should have mentioned that. You can treat the cells with sodium butyrate, the acetylation level shoots up, and at the same time, the methylation level goes with it. So essentially, the acetyl precedes the methyl, and that now makes sense based on charge accessibility. I do call attention to this, that this was known for years in one way. The relationship between methylation and acetylation has been understood for years. It's just been very difficult to actually demonstrate the pathway in activity like we've done. And so I do call attention to Dave Alice in Tetrahymen. I showed this some years ago. Dave Alice, an absolute giant in our field, who sadly passed away last year. And I know I'm going to enjoy reading Dave's papers in the years ahead, because Dave found most of the stuff that we're now just dissecting. So here what we're doing is looking at a Triton acidurea gel in Tetrahymena. We're separating the H3 forms based on charge, and then we're immunoblotting for K4 methylation. And what you're seeing is that the higher acetylated forms of the tail are the ones where the K4 trimethyl is localizing. So all of this has been known. All we've done is sorted out the order of things. Okay, but now once you've got your methylation and acetylation on the same tail, question. Remember, readers are presumably coming into play. So this brings us to a protein called BPTF. This is work we've done with Catherine Musselman. BPTF is a part of a 4-subunit complex called NERF, but at its C terminus, BPTF contains a PhD and a bromo. So we're just looking at 2 of the reader domains within this. Now we're looking at the ability, by the way, of the acetyl, not for charge neutralization, but the ability to actually be reader engaged. So PhD years ago has been worked out by Alex Ruthenberg in the Alice lab and others following on as a K4 trimethyl reader. The bromo has been a bit more controversial. There's people who say it binds to H3 and H4 acetyl. That is true on peptides. On nucleosomes, we see it as primarily a H3 acetyl reader. But if we create various nucleosomes, we should be able to see the interplay or the single engagement of the reader domains and the combinatorial engagement as well. So this, in fact, is the concept of multivalency writ large. One binding event is weak, but 2 binding events stabilize each other and should be much stronger. So the PhD should bind K4 trimethyl, the bromo should bind the acetylation, and together they should be able to bind a methyl and acetyl. Can you see this interplay taking place? On the nucleosome, you can. That's the unbinding to the unmodified. And remember, I told you the further you are to the left, the stronger you are as a binder. So that's the binding of the PhD to the methyl, the bromo to the acetyl, and that is the combinatorial engagement, about a log increase in binding. That's the histone code in operation, by the way. However, this is the really joyous part. If you go back to a peptide, you can't see this. One of the reasons perhaps why the histone code has been so controversial, because lots of predictions couldn't actually be identified when you went to the substrate that you were working with. Here you see the binding to the unmodified peptide. The red is the methyl, the purple is the acetyl, and the green is the methyl acetyl. So in other words, you can see the histone code in operation on a nucleosome, but not in operation on a peptide. So the physiological substrate has given you additional information. So, question, can you actually dissect what's going on? And is it in cis or is it in trans? So remember, there's 2 H3 tails. Is it actually binding on the same tail or across the 2 different H3 tails? Because we know how MLL is making it, and it's doing it on the same tail. So here we return to our heterotypics again. The unmodified in blue. Here we've got one tail is unmodified. The other tail contains K4 trimethyl. BPTF, PhD bromo can bind to that. Now one tail is unmodified. The other contains a single acetyl, because we turned it out to drill down to the single acetyl. That's not enough for bromo domain engagement. So you can see that there's no binding there. If we put the methyl and the acetyl on opposite tails, so the nucleosome is K4 trimethyl, K14 acetyl, but it's in trans, and what you see is no increase in binding. However, if you put them on the same tail in cis, now you get a 24-fold increase in binding. So its PhD bromo is looking for what MLL was creating. And to finally show that it is primarily a cis-driven event, you can create a homotypic nucleosome with both tails modified, and what you see is essentially just a slight increase in binding, just because you've got 2 binding events. So essentially the 2 binding events are essentially independent of each other. So we then sort of thought about what we would normally do. Is you would put together a publication, you would send it off, you would do genomic mapping of the methyl, the acetyl, and BPTF. And reviewers would come back and go, well, you're looking at a population. You can't definitively prove that they're binding to the same nucleosome in your genomics. We have this ability that we've publicized, and in fact, we encourage all of you to consider it. You don't need antibodies for cut and run to go after your targets. You can actually use reader domains to do it. So here we have the same biochemical reagent we had a moment ago, the GST PhD bromo. To turn this into a cut and run capable reagent, you simply need an antibody to GST. Because remember, cut and run works by recruiting protein AGMNase to an antibody. So this is the antibody in here, but the specificity is potentially driven by this. To prove that the thing was working, we created nucleosomes because that's what we do. And we've created a nucleosome that's unmodified methyl, acetyl, or methyl acetyl. We add that into a cut and run experiment. And what we immediately do is we go and look at the standards before we look at the genomic tracks. And what you immediately see is that it does not bind to the unmodified nucleosome. It does not bind to the acetylated nucleosome. It weakly binds to the methyl nuke. But what this reader, a cut and run, is actually looking for is the combinatorial signature. And that's exactly what it finds and enriches for. You can see it in the standards, and you can also see it in the genomic tracks. Here's a chase map version of it: methyl, acetyl, endogenous BPTF, or exogenous, the stuff that we're adding in. If we look at tracks, what we see is exactly what we hope to see. Regions that contain methyl and acetyl recruit endogenous and recruit exogenous. Across the absolute majority of the genome, that's what you see. You don't really see regions with methyl and no acetyl, and I've explained to you why. But what you do see are regions with acetyl and no methyl, and they don't recruit either the endogenous BPTF or the stuff that we've added in for our biochemical assay. And there's a subset of data in there, and so I also want to call attention to this when we're considering our experiments. The histone code is never sort of envisioned as this continuous addition of positive signals, one positive signal after another, all just driving stuff into the arms of that. There's also potential for negative signaling in here, and I call attention to this. It's a very small subset of the peaks, but it's regions where there's abundant methyl and abundant acetyl, which is very good at recruiting the stuff that we've added in, the reader cut and run approach, but it doesn't recruit the endogenous. And personally, I think what's going on is controversial. What I think is going on here is we're simply looking with this construct at this part of nerve complex. That's all it is, two positive signals sitting together. But the entire complex has a lot of other domains, and who's not to believe that there's a lot of there's a negative activity saying, well, there's something else at this location that nerve doesn't like, and therefore the complex will not be localized at that location. How are we doing for time? Okay, for the last 5 minutes, I'll sort of change to a completely different track, and when we started Epicipher and we started making our nucleosomes, we thought, naively in hindsight, we thought one of the groups that would sort of get most traction out of them would be things like drug developers, because they were trying to develop inhibitors of binding or inhibitors of an enzymatic activity, and they were using peptides, but there've been lots of data showing that the enzyme activity towards peptides versus nucleosomes were different. So we thought they would be the ones who would go the most for the nukes. Turns out not so much. They didn't use them for the initial screening, but they did use them for the follow-up. But over the last few years, the group that has gotten most engaged in the physiological aspect of things are the structural biologists. It's been amazing to see over the last few years, particularly when cryo-EM came on board, because it's now very difficult. If you work in chromatin biology, it's next to impossible, I believe, these days to create a structure in a high-profile venue of your reader, single reader, and a peptide containing your post-translational modification. That really doesn't get much traction anymore. Now with the advent of cryo-EM, everybody's pretty saying, well, you don't need to create crystals anymore, so let me see the pictures. So everybody wants to see intact complexes and intact nucleosomes. So that has been some of the most fun over the last few years, is seeing that in action. So I'm going to just show you some collaboration we did. It's a study, I think, that encapsulates everything, but it's a study that we did about a year and a half ago. It was published last year with Keogh Mash's lab, and it's of a deubiquitinating enzyme in associate with its substrate, K119 ubiquitination. So I just refer to ubiquitination. This is not the degradation stuff that's recruiting the proteolysis machinery. This is actually monoubiquitination, which is used as a signaling event. So there's been lots of monoubiquitinations identified on the nucleosome surface, enzymes, the reason why they're there, the enzymes that add, the enzymes that take away. I just call attention to this. Since they're at a given residue, they're actually at a given position on the nucleosome surface, and they're at a given position relative to everything else that's on the nucleosome surface. So therefore, that in theory can drive enzymology, because if I dock in the nucleosome in a certain way, I might only be able to reach one ubiquitination and not another. So therefore, an understanding of the structure might give me an understanding of specificity and enzymology. So I call attention to where K119 is, and you can see K119, which one is the most studied marks, is in a very different position on the nucleosome surface versus one of the other most studied nucleosome ubiquitolations, K120. So you can immediately see, based on structure, why you might have an understanding of the biochemistry and the biology. So this is the complex that KJ's lab decided to work on. It's BAP1 ASXL1. It's a heterodimer. So it's the PR-dub complex. BAP1 can form into heterodimers with 3 ASXL subunits, 1, 2, or 3. So we've just taken the conserved region between the 3 of them to create this heterodimeric complex. This was then assembled with a nucleosome, and the cryo-EM was created. Now, I'm not a structural biologist, so I'm not going to talk about the salt bridges, et cetera, et cetera. I'm just going to look at the pretty pictures. And what you can immediately see is when you sort of drill down, you can see that there are multiple points of protein-protein contact within the heterodimer wrapping around each other. Plus, here is ubiquitin, and here is the nucleosome surface. You immediately see a structure, as in it's not just this random event. It is touching at a specific place and orienting the ubiquitin, its target, within its active site. You can see multiple points of contact. You can see the contact with the DNA diode, contact with the acidic patch on the nucleosome, and also contact with the DNA exit point. Each of these points of contact it's important for the way that the nucleosome is docked on by the enzyme, but it also orients the enzyme towards K119 ubiquitin, such that it can't reach other. So in other words, it drives specificity. It's also an understanding of, you can actually mutate each of these points of contact and look at a loss of enzymatic activity. So here we're looking at a de-ubiquitination assay done by one of the scientists, an extremely talented scientist in our company called Susan Glore. And essentially we're looking at full enzymatic activity versus compromising this activity with cysteine to serine in the active site, so complete loss of catalytic activity. And now you see, if I make mutations to the acidic patch binding region, or the DNA binding region, or the DNA exit point, I can immediately see a loss or a almost complete ablation of catalytic activity. So in other words, it's touching down on inside this biochemical assay, it's touching down on the nucleosome to actually effectively remove the ubiquitination. Also, there is BAP1 ASXL1 in fact, BAP1 is mutations are classified as BAP1 tumor predisposition syndrome. It's one of the most highly mutated enzymes in epigenetic regulators that are observed in cancer. And in fact, if we look at these acidic patch binding DNA diode or ubiquitin patch, or essentially across the 2 subunits, disease mutations are actually compromising catalytic activity in a biochemical assay. So now you have an understanding of specificity. You also have an understanding of disease biology. And then finally, the thing that sort of seals the deal for me, and I hope to sort of communicate, is all of this is based on the enzyme and its physiological substrate. You can see the loss of activity towards its physiological substrate. Here is comparing a nucleosome and a peptide. And so here is the fully active enzyme, the catalytically dead one. And what you immediately see are these nucleosome contact points, which are driving the enzymology on a nucleosome substrate, have essentially no impact in a peptide context. So in other words, if you were working with a non-physiological entity, what you can figure out is that it's dull, of course, but you get no understanding of its enzymology, and therefore you also get no understanding about this sort of disease relevance of these cancer-associated mutations. So with that, I'll stop, take any questions that Ellen has managed to accumulate. I apologize for the technical difficulties we were encountering early. I call attention to all of my colleagues at Epicypher, who are an absolute joy to work with, and it's an extremely talented bunch. We have to start getting widescreen photos. There's more of us these days than when we started. We are fully indebted to our funding agencies, who, like the NIH, has been so generous to us to allow us to pay for all these programs and to develop these technologies and roll them out to you guys. So many different collaborators, but particularly for today's talk, Catherine with the BPTF study, also with the newly given Dr. Fuchs from earlier this year, Brian and Kanishk for the MLL study, and KJ for the PR-dub study. And with that, I'll take any questions. And thank you all for your attention.
Excellent. Thank you, Michael. We do have a couple questions. The first one's actually related to cut and run the assay itself. The question is about negative controls, or what we use as reference samples. So specifically, like, do we use IgGs? Do we include no antibody reactions or input? So what can you say to that question?
All of the above. So you're asking how do we control for the cut and run itself or for a given antibody?
What do we use as negative controls? And I guess what do we use as positive controls?
So, it depends on, here's the simplest way of saying, if we're working with, if for every time we do a cut and run experiment with the sample, we include negative and positive control reactions. So I've got one sample and I'm going to split it into multiple reactions, and that will be an IgG, negative control, because you need that to give you the background signal. And then the two positive controls we add in are K4 tri and K27 tri. And we do that because they give you a very different genomic distribution. So that tells us our sample worked. We also, in that experiment, throw in K-Metstat, the nucleosome standards, because that allows us to monitor on every single day the performance in the experiment. Plus, we also add in E. coli DNA as a carrier, but also to basically keep track of that for library prep, et cetera, and for normalization purposes. For every experiment for which we have a nucleosome standard, we add it. When we're going after novel targets like transcription factors, if it's a novel factor, we always go after, we bring in, and I know it sounds like extravagance, but it's extremely important because you're about to invest years of work in this factor. We bring in 5 antibodies per target and we check them all. So we will bring in 5 antibodies to TF unknown. We'll make sure that they're 5 different antibodies, so we get monos and polys. I know that we'd much rather use monoclonals for renewability, but sometimes there's not a lot of monos around, so we'll bring in different. We try and ensure that they are different antibodies by looking at the epitopes or species that they're raised in. And then the reason for 5 is because accidentally you could get peaks being yielded from antibody A and antibody B, but you can't tell which are the right ones. So what you're looking for are independent reagents giving you the same peak structure. I believe that addresses most. And then, yeah.
Excellent. And we have another really interesting question, I think. Does the binding of reader proteins to histone PTMs limit the ability of antibodies to bind these PTMs in chip or cut and run? So is any biologically relevant signal possibly being masked due to readers?
Yeah, interesting question. I don't think so. I honestly don't think so. I think the way that, because everything biologically has an off rate. And for example, the acidic patch is one of the busiest parts of the absolute genome. But we can actually get reagents that we can show binding to the acidic patch. We use that for certain reader constructs that we've got. In that experiment with BPTF, I was able to show you that I could localize a BPTF signal. I could localize I think it's something in mind, but it's not something that we've never been able to track down, for example, a loss of signal allocation because, the factor is just so abundant there, it's masking the target. Conceptually possible, but practically we've never seen it happen.
And we also have a question about heterotypic nucleosomes. So any thoughts on or insights on heterotypic nucleosomes, for example, with a lysine ME2 and ME3 states on 2 histones? So I guess making a K4 ME2, K4 ME3 in terms of different reading to homotypic states for each.
I'm not sure I fully understand the question. So thus far we've managed to make heterotypic H3s, where the 2 H3s are differentially modified, and then exploring the biology there. That one's straightforward. I think we can do it up. I think we've done it. We could do it up to position 36, but I don't know how much further we've gone. We're exploring currently heterotypics at other histones on the N termini and also on the C termini. Those are R&D projects, but the H3 tail is completely accessible now to other projects. So one of the most interesting ones there, and it's in fact what Beat had published on, is bivalency. What about bivalency at K4 trimethyl, K27 trimethyl, which is a subset of heterotypics, because that biology is supposed to exist. So here you've got a nucleosome with a positive mark and a negative mark, except they exist on the opposing tails. Mm-hmm. So we've managed to make that nuke, and now we're trying to map its distribution with various approaches, us and colleagues. I'm not sure I understand the K4 dye try question.
I think what he was meaning was a heterotypic nuke where, let's say it's H3K4, and one of the histones has H3K4ME2, and the other histone has H3K4ME3.
Yeah, that's absolutely, that's no problem. I mean, that's straightforward. There's nothing that stops us making that. We haven't made it yet, but there's nothing that stops us making. I think we've made 14 or 15 of them for 3 different biology programs, just to see how they behave themselves. But yeah.
Nice. Let's see, another one about reader proteins. Are readers, besides lysine methylation readers, generally affected by tail acetylation? If that generally affects tail accessibility? So is it just something that is specific to like histone H3 tail methylation?
Yeah, I don't know. We haven't checked. A variant of that question is asked a lot. Not specific. So I'm trying to think. The only other thing that we've studied on the H3 tail is phospho. Let me rephrase that. We've studied on the H3 tail acetyl, lysine methyl states, arginine methyl states, and phospho, for example, serine 10 or serine 28. And I don't know if I might imagine that serine 10 is more accessible or other things are more accessible. One thing that I'm asked a lot is, well, you looked at K4, and so for K4, and you've shown that the K4 trimethyl goes with acetyl, and in most people's minds, acetylation neutralizes charge and it makes it available, but then acetylation is not generally associated with repression. But question, does acetylation drive K9 or K27 methylation activity just like it drives MLL, which is a positive? And the answer is there have been a few biochemical experiments done on K9, and yes, the K9 methyltransferases are driven by prior acetylation on the tail, and that's biochemically. The only problem is if you do PTM mass spec data, and Nick Young has done this, and others in fact, there's no relationship between K9 methylation and acetylation, at least not to the same degree as we've seen in MLL. So conceptually it's possible, but the enzymes are subject to additional layers of regulation in vivo that we're not seeing when we're playing with them in a biochemical setting. We have tried some work on K27 methylation directly in-house. As far as we can tell, PRC complex isn't driven by, at least not meaningfully driven by acetylation. So this charge neutralization thing is obviously working. I mean, it truly is, but the question is how it's controlled in vivo as well. It's not just a one size fits all.
So many questions are coming up now. Okay, I'll just kind of take them in order. So there's a question about the robustness of the assay, I'm assuming cut and run, with respect to heterogeneity of tumors. I'm assuming this may be related to the sensitivity.
Those are two very different questions.
Yeah.
But in terms of the robustness of the assay, we always do technical reps. We normally do tech reps, biological reps. Particularly for if you're doing a drug treatment or something, we would always recommend biological reps. Duplicates, yeah, triplicates occasionally. The assay itself is insanely robust. I mean, I'll be fair also, we particularly when you do it automated, because we're doing it on the robot, because then you've got 96. The robot doesn't get distracted. The robot doesn't get bored. But it is a very robust approach. The heterogeneity of tumors is sort of driven by sensitivity. So if you've got a mixed population of 500,000 cells, the question is, you're not looking at cell lines anymore, but you're actually looking at, well, you might have a cancer cell and a normal cell. In fact, we can actually see that. And in fact, we've done things like you can take a drug, you have a patient who's got a blast count. And so therefore with that, when you look at the feature maps of K4 methyl and K27 trimethyl, what you see are positive and negative signals lying on top of each other, which you don't normally see. And the reason is because things like proto-oncogenes are active in the cancer and not active in the normal cell. And in the mixed population, you see these populations on top of each other. But then over the course of the drug treatment, the cancer is being removed from the situation, hopefully, because that's exactly what the cancer drug is supposed to do. And now what you see is what appears to be the reestablishment of the normal signal. It's not because the cancer has moved to the normal signal. It's because the cancer cells are dying. And now the native signal is predominating. So there is you're seeing the heterogeneity change. Yeah, robust. Yeah, this is an insane. You need good antibodies. And this comes back again to why specificity is only one part of the question. If you've got an antibody that's only pulling 1% of what it sees, you've already capped the capabilities of your tech. So for us to get past this, even if we find a good antibody, we always check it with decreasing cell numbers, with cancer lines, which have a lot of everything, and native tissues if we can get them. And then we're really trying to identify not just an antibody that's eking out a signal, because doing those experiments is tragic. What you're getting is an antibody that's giving you robust signals so you can consistently do your experiments and you can start to trust your data a lot more. Another reason why you invest more time at the beginning screening antibodies rather than simply finding one and running with it. You're about to invest years of your life in this program. Get an antibody that works and then buy as much of it as you possibly can, because when the lot changes, the activity changes as well, or the capability changes as well.
Absolutely. And we're a little over time, but I think there's one more question I'm going to ask because it's a great question and it's one we get all the time at EpiCypher. Any thoughts on when cut and run or cut and tag works best in your hands? Their sense is that cut and tag behaves better for activating PTMs and cut and run is the way to go for transcription factors and heterochromatin targets, but they're not sure if that's anything beyond their hunch.
It's a hunch. It's wrong. We use both, and we've brought in both technologies. We have played with them. We've taken the initiating protocols that were put online, and we've continuously refined them. We continue to refine them. We try and automate them. The whole thing. Cut and run in-house is the workhorse technology. Everybody gets it to work first time. It works for pretty much everything. Cut and tag, it has its applications. Some people think it's an evolution of cut and run. It's not. It's a sister technology. It has the advantage, and it's a very powerful advantage. I mean, it's a bit faster because you don't need to do library prep. You're doing tagmentation and direct amplification. But in theory, not so much in practice, but in theory, you can change the tagmentation sequence and therefore do things like mix samples. And there are loads of papers, but they are, to be honest, rarefied approaches. Most people don't need those capabilities. Mm-hmm. It suffers, however, in a few ways as well. Cut and tag is trying to be ATAC. It's protein AG TN5. And so in an ATAC experiment, you throw NTN5 and it finds an open chromatin region, touches down, and it tagments. That's great. When you add in protein AG TN5, the protein AG is trying to find the antibody, but the TN5 is trying to find the open chromatin signal. To stop it becoming ATAC, what you do is a high salt wash. And the first thing that does is it displaces weakly bound factors. So in-house, we would never use tagment or TN5, or sorry, cut and tag for TFs. I know that there's data out there.
Erica actually, I think Erica was actually correct, and maybe I flubbed it when I asked it to you. So she said their sense is cut and tag is better for PTMs, and cut and run is the way to go for TFs.
No, even that. I mean, yes, it's true that cut and tag does sometimes give, because the high salt wash also gives you slightly cleaner data. But we've got lots of PTM data with cut and run. In fact, everything we do, we usually take a look at both. Let's just say that the advantages of cut and tag still wouldn't tell us to move in that direction for PTMs. There are too many additional benefits for cut and run. Mm-hmm. And again, I've got no axe to grind. We work with both tech. We see the strengths of both tech. We see the application of both. Personally, it's usually our go-to. There's a special reason, personally, why we would use cut and tag.
Fair enough. Thank you, Andrea Johnstone, for answering a bunch of questions in the chat, by the way, and Carolina, our
my colleagues are cleaning up around me as well. Yeah. This is great.
Yeah. This is fantastic. If we didn't get to your question, we will follow up with you. I think Andrea actually addressed a lot of them. Thank you so much for joining today. We really appreciate it. Any final words, Michael?
No, this has been fun. Thank you, Ellen, and for helping to organize, and the Livestorm crew. as well. Is technically, obviously, the bit in the middle, but I think that was well, that was entirely their fault. But no, I don't think it was their fault at all. I think it was just some screen selection choice. This is great fun. I mean, anybody who wants to reach out, we're out talking all the time. My colleagues are doing so many stuff that I didn't talk about today. If you want to know about lysine methyl acetyl, any PTM on Earth, pretty much ask us. We've almost certainly done work with it. And we're also doing work on DNA methylation, open chromatin, big work on FFP. Automation is fantastic. The speed at which we can do things. We'll tell you basically about anything you want to know unless it's collaboration, in which case we won't. But thank you all for your attention.
Thanks. And like I said, this will be online afterwards for anyone that joined late or wants to see it or share it. So stay tuned. Thanks, everyone.
Thanks, everybody. Bye-bye. Have a great day.